Okay, everyone, we are starting our evening session, and it's my special and great pleasure to introduce Dr. Rudolf Valenta from Medical University of Vienna. And this is a special of Sochi Dialogue, the collaboration among Russian and Austrian scientists, specifically in the field of biomedicine. But Dr. Valenta has even more specific relation to Russia because he is a head of one of the laboratories built through the Mega Grant program. And he has a long collaboration with Russia and specifically managing of a Mega Grant-based laboratory for the last few years. So it's quite interesting for us to get his feedback and vision on science and collaboration with Russia and strong and weak parts and maybe some advices and suggestions how it can be improved, updated, and what target of research should and can be chosen. Thank you very much. And Rudolf, please start.
Yes, thank you very much for this very kind introduction. When you ask me about weaknesses, I can say there are almost no. It's a pleasure to collaborate with Russia, and it's very easy for people from Austria. And in fact, it's very simple because you can fly Moscow, Vienna several times a day. It's a short flight, so we are actually very close together, almost neighbors. And it's therefore really a pleasure and easy to collaborate with Russian scientists. Now, as you mentioned rightly, the work that I'm going to present to you has been achieved in the course of a mega grant that was conducted at the Institute of Immunology in Moscow, together with Professor Musa Kaitov at the Institute of Immunology. And I would like to present you now the fruits of our work, and I think they may be quite relevant, as you will see in a minute. They concern the development of a vaccine for the treatment and prevention of birch-borne allergy and a very special syndrome that is the oral allergy syndrome. That's a food allergy that is associated with birch-borne allergy. Now, I think everybody knows what a vaccine is, and usually we use vaccines to prevent the disease, like we develop vaccines for SARS-CoV-2 to prevent COVID. Now, in the case of allergy, it's interesting because it's a little bit the other way around. We are using a vaccine for therapeutic treatment. So this means we can use vaccines in both ways. On the one hand, to prevent diseases. On the other hand, to treat diseases. In allergy, we treat. And the thing that is the novelty is it has not yet been used to prevent allergies. But I'm also going to show you some results on this. Now, I think everybody knows somebody who has allergy, and this is because allergy is a very common and very frequent disease. The most common manifestation is the hay fever. It's allergic rhinitis and conjunctivitis, but allergy can be also life-threatening in the case of asthma when it affects the lungs. It can produce gastrointestinal symptoms. It can kill people by systemic anaphylaxis. And it can create awful skin manifestations like atopic dermatitis or urticaria. So allergy affects many different tissues in the body, many different organs. And it affects, and this is the important thing, many, many people. We can count that about 30%, if not more, of a population is affected by allergic symptoms. So I think it's a very important disease that we have to cope with, because it can be life-threatening and it can be really severely reducing the quality of life. Now, the things that happens to an Austrian, when an Austrian travels to Russia, the Austrian will see something like this here. And this is a forest of birch trees. You can see it when you drive from the airport in Moscow to the city, but you can see it also when you are, for example, traveling in the Ural, for example, like here from Ekaterinburg to Tobolsk. And then when you work in the field of allergy, you may guess that birch is perhaps one of the most important allergen sources in Russia. Now, to be allergic to birch means that you react hypersensitive to something that is in the birch pollen grains, that are spread in the air in the spring. And what is in the birch pollen grains? It has a name. It is a specific molecule that has the name BETV1 Betula verrucosa number 1. It's the major allergen of birch, and we have actually cloned this allergen in the late 80s in Vienna. And I have developed it here now in the mega grant program further as a diagnostic biomarker for identifying patients with different phenotypes of birch pollen allergy, which is published in this, I think, nice work by Olga Elisyutina that we produced in the mega grant program. What we do here is we use these molecules, the BETV1 allergen, and you see here related allergens. And with related allergens, I mean allergens that are very similar in the three-dimensional structure in the sequence to BETV1. BETV1 is dominating the sensitization. You can see that more than 65% of allergic patients in the Moscow region react with the major allergen BETV1. They also show reactivity, for example, with an allergen that is present in apple. So people who are allergic to birch very often have also allergy to apple, but also to hazelnuts, to peanuts, and to other foods. So there is cross-reactivity. Now, you can assess actually the IG binding to all these individual allergen molecules by chip technology, as we did it here. And you can also measure the levels of BETV1 specific IgE. And there is in fact something very interesting in birch pollen allergy. There's people who have allergic symptoms like in the upper panel, and there's people who have not yet developed the symptoms, but they have what we call a silent sensitization. It's like a precancerosis that you only would detect by laboratory investigation. Now, in people who are non-allergic but have already the IgE, you will see that the IgE levels are much lower. The other important aspect that you can see here is that there's different phenotypes of birch pollen allergy. People with allergic rhinitis, with allergic asthma, with oral allergy syndrome. This means when they eat, for example, apples or hazelnuts, they get swollen lips and swollen throats, atopic dermatitis, and the silent sensitization. What is very interesting here is that those who have also the food allergy, due to cross-reactivity with the BETV1 allergen molecule, are the ones that have the highest BETV1 specific IgE levels. So we can use BETV1 as a surrogate marker, biomarker for food allergy to identify actually these patients. Now, the next thing is when you work systematically to cope with a disease in a country, is you have to learn more about the country's sensitization profile and what is the need in a particular country. And for this purpose, we have basically started a study where we studied in Russian children IgE sensitization profiles. And for this, we use a technology that is called microarray technology. It's basically a chip that contains immobilized hundreds of different allergen molecules that were produced by recombinant DNA technology. In order to investigate this, we have enrolled children in the Moscow region, 103 with symptoms of allergy and about 100 without symptoms, but yet we found also sensitization in them. Now, when we assess the allergic children and we test them for all the allergen molecules that are basically listed up here on the x-axis, as you can see here, and we measure the percentage of sensitized subjects, we can see that at the peak of the sensitization, most of the children, as I said, more than 65%, are sensitized to the one BETV1 allergen molecule. It's the 117 kilodalton protein. Very interestingly, you may note that the second most common molecule that is recognized by allergic people in Russia is the major allergen of cat, Felis domesticus number 1. And if people have read Pushkin and they know Pushkin, people will know that basically in many Russian homes, there's also cats. And cats produce this FeLD1 allergen, and it's a very important trigger of asthma. So there's two allergen molecules that are important for the Russian population. It's BETV1 and it's FeLD1, followed then by some of the others. And you can see nicely that with all these molecules, you can find out what is important in what region. If we would look, for example, in Sochi region, in Krasnodar, we would find ragweed more prominent and perhaps also mugwort. So there may be regional differences. And the chip diagnosis actually allows you to find out what is important in what part of the country. Now the hit list can be displayed also in a picture like this. BET-V1, the most important allergen, followed by the major cat allergen, FeLD1, and then comes the other ones. Now, I think establishing such profiles is very important if you want to go for the vaccination, because you want to know what is your enemy. What is the most dangerous and the most common enemy you have to fight against? And here is BET-V1 and it's FeLD1. When we look then in food allergen sensitization, we can easily recognize that the food allergens that play the most important role in Russia are those that cross-react with the BET-V1 allergen, present in hazelnuts, in apple, but also in peanuts to some extent and in different fruits and vegetables, whereas others, like for example egg or fish, is much less common. And these actually cause the oral allergy syndrome. Now the idea in this project was to develop a vaccine that will protect you not only against the birch pollen allergy, but also against the apple allergy and the food allergy. And for this we have used a very new technology that we developed recently. And you can see here a molecular engineering of such allergy vaccines. There's different types. And the most recent, and I think most successful, is the one that we have applied in this mega grant project. It basically uses a carrier protein to engineer the vaccine, the active ingredient of the vaccine, that means the antigen that we inject into the people. And this carrier molecule is very important because it promotes the immunogenicity, so this helps us to get a strong production of protective IgG antibodies. You could use the same technology, by the way, also, of course, for developing of COVID vaccines, and in fact we do and are very successful with this. Now, in the case of allergy, the engineering of the vaccine is a little bit more complicated than even making a COVID vaccine, because we have to avoid side effects that are caused by IgE recognition of the allergen, because of course injection of an allergen molecule into an allergic patient will cause allergic inflammation. So we have developed a technology where we fragment the allergen and then put the pieces of the allergen to the carrier molecule to avoid IgE activity and T cell recognition, to bypass the side effects in the course of vaccination. Now for BET-V1 and MALDI1, we know where the IgE binding sites are on the molecules. This is the BET-V1 sequence on the top, followed by the MALDI1 sequence from the apple allergen. And similar amino acids are displayed by dots. And basically these two peptides indicate a surface on these allergens where IgE antibodies of allergic patients like to bind. You can visualize these binding sites, although also on the three-dimensional structure of the allergen. This is the binding site where IgE likes to bind on the BET-V1 allergen. It's on the front side. When you turn the allergen around, you will see it's not on the back side, and the same is true for the apple allergen. So there's one prominent recognition site, and we take advantage of this knowledge, take out the peptides, and use it for the construction of the vaccine. Now today, vaccines are not made anymore by shaking pollen from the trees, like we do not produce viral vaccines by cultivating virus. We're using modern technologies, and in our case, we create subunit vaccines. And in our case, we use as a carrier protein one particular protein that is also deserving some interest. It's the pre-S protein from hepatitis B. The reason why we choose this protein as a carrier molecule is not disclosed in this report, but I can tell you very quickly. It is because this protein is used by the hepatitis B virus to dock onto the liver cell. And this means that when we create such a vaccine and incorporate into the pre-S also the epitopes of the allergen, we get a combination vaccine that will not only protect against allergy to apple, birch, or both of them, like in the combination construct, but also against hepatitis B. So just bear this in the mind. So this is showing you what you can do in a creative manner by engineering new vaccines. Now you can see that these proteins that consist, and these are completely artificial synthetic proteins, out of the pre-S and the epitopes of the apple and the birch allergen, can be very nicely produced in Escherichia coli by fermentation. So they can be produced in bioreactors in large scale in 100-liter amounts and in gram amounts for vaccination. And the proteins are quite easy to purify for the final formulation, where we use aluminum hydroxide. I mentioned to you that engineering allergy vaccines is more complicated than even engineering vaccines for viral infections, because you have to pay attention that the vaccine is hypoallergenic, and this means that it should not induce allergic reactions upon injection into the sensitized patient. We test for this by checking the vaccine constructs for IgE reactivity. The allergen molecule BETV1, as you can see, binds IgE to a high extent in sensitized patients. However, the vaccine constructs that we create are kind of attenuated, and this means that, like here, the EBPS protein does not bind IgE antibodies, whereas BETV1 binds IgE antibodies. So this is a hypoallergenic protein. You can show this also by a cellular experiment where you show that basically the vaccine constructs do not trigger the release of histamine and biological mediators from mast cells and basophils, whereas the allergens BETV1 and MALDI1 do so. So this is a very first and important test to show the safety of the vaccine before you come into vaccination of patients, in this case for therapeutic vaccination. Now we are competitive in our project and we wanted to make the best vaccines in the world. So we benchmark our synthetic vaccine against all the known allergy vaccines that are produced in Europe, like in Denmark by the company ALK, like in Germany, actually by the company Allergopharma, Rheinbeck in Hamburg, Allergovit, but we also benchmark ourselves against the Russian product, it's Microgen. What is the difference? The difference for Alutard, for example, is that patients have to receive multiple, more than 20 injections. With Microgen, the Russian product, you give more than 30 injections before the pollen season to protect the patient. Our recombinant vaccine actually needs only 5 injections before the season, and then before every birch pollen season, just 1 booster shot, so it's very simple. You can see already that actually treating allergic patients has something in common with making vaccines against viral diseases. Now, how do we benchmark? We measure actually the induction of protective antibodies in vaccinated animals. And in our cases, we use rabbits because they are outbred like humans. We vaccinate them with the different vaccines, Polinex, Alutard, the Danish vaccine, Allergovit, the German vaccine, Microgen, the Russian vaccine. And then we measure the extent to which the IgG antibodies induced in the rabbits inhibit the binding of allergic patients' IgE to the BETV1 allergen. You can see that Allergovit seems to be the best product with natural allergen extracts. It inhibits the binding of allergic patients at around 60%, 55 to 60%. However, the recombinant vaccines that we produced in the Megagrant project are more potent. You can see that we achieve more than 75% protection actually in this assay, in the blocking assay. And this means that our product is significantly better than the best available extract-based vaccine from Germany, the Allergovit. So we are proud of this. Importantly, our vaccine can do something that the other vaccines cannot do. In fact, our vaccines can actually induce also IgG antibodies, protective antibodies, not only against BETV1, but also against the major allergen of apples, the MALDI1. And you can see here that the combination vaccine that contains the epitopes of also the apple induces the best inhibition of IgG binding to the apple allergen, better than the one that uses only the birch pollen peptides. So we are superior here, and by that we can hope that we can protect our patients not only against the birch pollen allergy, but also against allergy to apple and also hazelnuts, as we found out. Now this is, you can say, quite a nice achievement. We have succeeded to construct a fully synthetic recombinant vaccine that is hypoallergenic and thus safe for allergen-specific immunotherapy of birch pollen and associated oral allergy syndrome. It's superior to all registered allergen extract-based vaccines that we could find. And in fact, we are ready to go with these vaccines very soon, actually in clinical trials. Now, this is the treatment part, but now let's be scientists and let's dream. Let's dream about what else can we do and consider also the peculiarities of Russia and the advantages that this country brings for us. Now we come here to a completely unknown territory in the science of allergy, and this is the making of preventive vaccination against allergy. Mission possible, we ask here, and I explain you how we want to do this now in the next steps of this project in Russia. Now there is possibilities for preventive vaccination in the following scenarios. When a child is born and is exposed to allergens, those that are prone to become sensitized will develop IgE antibodies. So what you can think is that when the child is sensitized and has not yet symptoms, you can test with the test that I showed you before for the presence of BETV1 specific IgE antibodies. And then what you can do is you can do a secondary preventive vaccination. That means you vaccinate a child that is not yet sick. And the idea behind this is to prevent the child from becoming sick and developing severe symptoms. So it's like in cancer, early diagnosis by a biomarker and then treating the biomarker to prevent the development of symptoms. We call it secondary preventive vaccination. And this should be actually possible. How can it be made? The study here shows you how it can be done. It's a study from Sweden, a country that is exposed to birch pollen to a similar extent like Russia. And you can see that in Scandinavian children in Sweden, at 4 years, 8 years, 16 years in a birth cohort, there's an increasing prevalence of sensitization to birch pollen allergen BETV1. 25% of Swedish children in the Stockholm area have IgE antibodies against BETV1 allergen. Now this can be measured by the technology that we have developed. And there's something very interesting. When you look now in these children, and you look at the children that have only a BETV1 specific IgE in the pink part of the Venn diagram without symptoms, you see that only few of the children at the early age, at 4 years, have symptoms. They are displayed in the cross-hatched and in the white Venn diagrams. The older the children get, the more of the children show symptoms. So we can clearly see that with every year of birch pollen exposure, the children progress towards symptoms. It's like a cancer that is growing in the child. Now with our tests, we can basically do a very simple thing is we can go into schools if we want, or in kindergartens, and we can measure BETV1 specific IgE levels at the early age, like 4 years here in the Swedish cohort. And taking a certain level of BETV1 specific IgE predict the incidence of the development of allergic rhinitis in the children at 8 years. This means we have a prognostic test that allows us to identify children that are at a high risk for developing symptomatic birch pollen allergy. And by that, we can define the children that deserve the secondary preventive vaccination. Now let's go in our minds a step further. It's like going to the cosmos. There is more to achieve, and this is primary prevention. Let's think about something very simple. We have many childbearing women in the country. And in fact, there is something interesting that we find is when women develop high levels of allergen-specific IgG and they transmit these IgG antibodies to the children, what we see is that these children are actually protected against allergic sensitization. And it does seem that when mothers transmit high levels of allergen-specific IgG, they can protect the child from sensitization. So the idea in this primary preventive vaccination scenario is that we can basically vaccinate childbearing women in the childbearing age to produce high levels of BETV1 specific IgG. And then we can boost up these IgG levels in the third trimester to actually make the women to propulse to the child the protective IgG antibodies. Now, this is actually a very nice idea, and that can be studied because you have seen in Russia, like in Scandinavia, we have almost 25% of the children that get birch pollen allergy. So we can relatively easily actually perform this trial, and it should be safe with these new types of vaccine. And this, in fact, would enable us eventually to make Russia the first country that can be eventually completely free of birch-borne allergy, and if the model works, of course, also of other allergies. Then, of course, we have another scenario, and this is early vaccination of the child early after birth to induce the protective antibodies early in the child to prevent the sensitization. And this is, of course, the most delicate one, and this is why we propose in this overview article a stepwise procedure in our scientific approach, where we first check the safety of therapeutic vaccination in allergic patients. This is where we are with our vaccine now. Then prove the safety by vaccinating non-allergic adults to look if our vaccine does not sensitize. This is a very important safety step. We have evidence that this actually can be fulfilled. Once this has been shown, we can move the next step to the secondary preventive vaccination and eventually prenatal vaccination of mothers, and then to the early postnatal vaccination. And if we run this program, as I say, Russia can be the first country without birch-borne allergy at all by a preventive allergy vaccination program. Now, I would like, before I extend my greetings, remain in this scenario and say that actually the ability for me to come to Russia has also allowed me to work with other Russian institutions, and like for birch, we have built up also in the meantime a vaccine for treating of cat allergy with Sechenov University. So we have basically targeted the two major allergen sources in Russia, in the Russian Federation, and we are ready to go now into vaccination approaches. And for me personally, this has been a very fruitful collaboration because it allowed me to develop further my dreams and to work with enthusiastic colleagues. And this is why it comes from the bottom of my heart that I send my greetings to my colleagues in Russia from Vienna. All the best. And the only reason why I cannot be with you is not COVID, because I have been traveling despite COVID. It's only because my mother has got severely ill and I have to keep her home and take care for her at the moment. Otherwise, I would be with you immediately in Sochi and spend the time with you. So thank you very much. And I shall be happy to take questions if you have. Thank you very much.
Rudolf, thank you very much. Very informative talks. These days, vaccine topics is so hot. So I think people are so much involved in that. So if you have any questions, comments, please give it. Or let me start. Rudolf, so let me ask you. These days, people are so concerned about potential side effects of vaccines. And birch vaccine is a kind of not so common in our, not in front of us. So how safe to get extra vaccine and so make it like Russia make a first country to be free of birch allergy. Yes. So what is a kind of balance of safety and effectiveness and with public concerns? How do you feel, especially working in Russia? I think you heard a lot of comments and opinions from common people and from your colleagues.
Yes. No, this is exactly why we work on this type of vaccines, because to our knowledge here, these type of vaccines that we develop here are the most safest that you can make. They, you have seen, they are basically non-allergenic at all. So it's a new technology, and I would rate it really the best and the safest. So the concerns are very low. And what is the nice thing is I always say, give the people the thing and make them believe by testing. And it was very easy. I give it in the hands of the Russian colleagues. They test it and they can see what it is. Yeah. And everybody is happy with the safety profile, at least in the cellular tests. We will then move to the skin tests. It's very easy to do. So it's the most safest technology. And of course, I understand the concerns. Because we have now a different type of generation of vaccines. And this is, I call it genetic vaccines. You will see that our vaccine is a recombinant vaccine. The safety is, of course, here also especially high because we inject the antigen. It remains deposited in the tissue in a depot. It's not spread like the proteins after genetic vaccination. So we have here an excellent safety profile, and I think we are ready to go these steps, but you see we are walking very, very carefully, of course, because the safety is the most important. So the scientific ambition has to be always balanced by the concern. And I work as a medical doctor, and this is very close to my heart.
Thank you very much. Any other comments? Because I have a following question. Rudolf, so we are talking about kind of practical aspects of working in international teams. Specifically, the Sochi Dialogue is a kind of Russia-Austria part. So tell me, so like a provocative question, but what is the hard part to work with Russian colleagues, and why would you advise not to work in Russia if there are any kind of
I'm sorry, I cannot give you this advice because I love to work with Russian colleagues. Yeah, so it's kind of this is I never encountered this problem. Yeah, quite the opposite. I enjoy working with Russian colleagues because I feel that we Austrians and Russians share many things in common. We like to go to Banya, we call it sauna in Austria. We like the forests, we like nature. We are natural people. Russian people are very outspoken and very direct. I like this. It's very helpful, yeah, in fact, and it's very productive. And I can say the most important thing is that, of course, you have to bring young people from here and there. And I have in my lab here in Vienna several Russian students that have made their career. You will not notice it in the publications. And I love to work with young talents in Russia. And there's one special thing is young people in Russia are very hungry. They like to work, and they do not always look at the watch here, so to say, and just sit there and do nothing. So they are enthusiastic, and I can only say the best.
Thank you very much, Rudolf. And maybe if no other comments, but questions. So, you know, these days, Russia trying to find their strong part in biotechnology and the so-called gene technologies. This is
Yes.
a big program of gene technology development. So in your sense, it sounds like your experience in practical implementation of innovative vaccines was very much successful. And also COVID-19, the Sputnik V vaccine is also kind of getting more popular and appreciated.
Yes.
So how do you think this vaccine development area, is it one kind of strong part of Russian biotechnology? Should it be further promoted? It can be kind of internationally recognized. What's your opinion on that?
Yes, absolutely. No, I have also experience, of course, in visiting different plants. So I know that in Russia you have the capability to manufacture vaccines, different types, of course, vaccines produced in E. coli. I've seen the plants, I've seen the people who can make it. It is there. I think what you have to do is you have to develop a very good vaccine candidate. The production is the less difficult one because the procedure is known. And it can be shared with the colleagues. It's like a ABC, it's a call it a SOP that you can follow. I know it very well. And I think a country is very well advised to invest in vaccination for a simple reason, because you will see that in preventing a disease by vaccination, you can do so much for the health of the people in your country, much more than when you can do when they are already sick and are very ill. And I think it's a good idea to spend investments and thoughts into developing vaccines for viruses, for viral diseases, also for allergy and other diseases. I think it's a very important thing, and you have the infrastructure, you have the people, and I think you have to have only good vaccines, and that's also important.
Thank you very much. So let's thank our speaker again. Thank you. And yes, and my proposal is just to catch up with our schedule to move to the next speaker, because it's in the same direction and the same vision, I think, in the field of immunology. Another very good and active collaborator with Russian scientist, Dr. Winfried Franz Pickel from the Medical University of Vienna. And we are going to talk about, yes, this is quite an interesting topic about education and research and PhD program in the field of allergology. Thank you very much. And please, Dr. Pickel.